from cDNA and PCR to CRISPR. 3 other powerful biotechnology tools should be briefly mentioned here: cDNA, PCR, and CRISPR. Complementary DNA technology, cDNA, is now routinely used for analysis of messenger RNA sequences, the processed mRNA sequences that are ultimately translated into protein sequences. cDNA uses an RNA template to synthesize a DNA sequence, which can then be cloned with recombinant DNA technology, sequenced, or used in any other way. In 1987, Kary Mullis invented PCR, which earned him the Nobel Prize. PCR is an extremely powerful method that is much faster, easier, and less expensive than recombinant DNA technology. Today, PCR is a multi-billion-dollar industry. The technique is routinely used in both basic research and for a variety of clinical applications, as well as forensic analysis, paternity, and to determine human ancestry. It can be used to create large numbers of copies of specific DNA or RNA sequences, and all that is required is that about 40 to 60 nucleotides of the flanking sequences of the target DNA sequence be known in advance. This means that virtually any target sequence of DNA can be amplified, copied in large numbers, even from total genomic DNA. The technique is so sensitive that the genomic DNA of even a single cell can be used as starting material. PCR now plays a major role in evolutionary DNA sequence comparisons used to determine relationships between distantly related organisms. CRISPR is a very powerful and new technique for targeting and changing specific DNA sequences in genomic DNA of living organisms. Human beings can now artificially alter the nature and evolution of various species, including humans. CRISPR has been quite useful as an experimental tool and has great promise for advances in molecular medicine. But there are several reasons to be concerned about ways that this technology could be abused to create designer babies and to destroy the natural balance of the human gene pool. Unfortunately, human greed, rather than human need, is often the driving force in biotechnology. Powerful technologies such as CRISPR could profoundly impact the future of our own species and the planet that sustains us. We need to be proactive in our decision-making to assure positive outcomes.